because it is one of the genes that's reported as being associated with prostate cancer. Not only being associated with prostate cancer, but being inversely associated with more advanced prostate cancer. This is being reported as one of the aggressiveness genes for prostate cancer, as a protective genetic variant. And there has been, with regard to its role in prostate cancer, there's been a lot of debate. I think people are in agreement that this is the gene that causes your PSA to increase. It may be more likely associated with just PSA levels as opposed to prostate cancer. But why would it be associated with aggressive prostate cancer? Inversely associated. So it increases your risk of prostate cancer, but decreases your risk of aggressive.
If you have high PSA expression, you're more likely to be screened. Or like
That's exactly it. So, but what happens if you're more likely to get screened? You find it early, right? And so that explains why it's associated with, because that gene is getting you into the screening paradigm, which is finding the early disease. Okay, so what we ended up finding, we found about 40 different variants that are associated with PSA levels, right? And that were replicated across all of these different replication populations. So the second thing we want to do is get into this, what I was hinting about with the KLK3 PSA gene. And that is trying to separate out whether or not it's associated with PSA levels or with prostate cancer. So this little DAG will hopefully help you understand how complicated it is. So if we could have a single nucleotide polymorphism in a gene or a gene region, well, that could be associated with PSA levels, or it could be associated with prostate cancer, with latent prostate cancer. Right? Either way, though, whether it's associated with prostate cancer, which then affects your PSA levels, or it's just associated with PSA, either of those are going to get you into the screening and having biopsy. So you can see right here, and then you end up with kind of biopsy-confirmed prostate cancer, that it's really hard to disentangle these two things. How do you actually separate out whether or not a genetic variant is associated with screening versus disease? So one thing that we did was we actually just compared the p-values and the regression coefficients for the genes and the SNPs that were associated with PSA to those that were associated with prostate cancer. So in this diagram, the blue SNPs here, okay, so what we have here are the minus log base 10 p-values for PSA going out in this direction, and then the minus log base 10 p-values for prostate cancer in this direction. And the blue dots here are all of the 100 plus variants that have been associated with prostate cancer, where the red ones are the ones that that we detected here that maybe were associated with prostate cancer previously, but were showing up also in our study as being associated with PSA in men without prostate cancer. And the black ones are actually, okay, so we've got actually, I'm sorry, the black ones are the ones we already knew about. The red ones are the novel hits that we have found being associated specifically with PSA that hadn't been reported for prostate cancer. So you can kind of break this. This is all in the Kaiser data, and you can kind of break this into 4 different quadrants. So out here in the upper quadrant are the things that are associated with both PSA and prostate cancer. And then over here we have things that are only associated with prostate cancer, as opposed to over here, things that are associated only with PSA levels. So you can see there's actually quite a few out here that are just PSA, and here's the KLK. We actually have an arrow on it because obviously the p-value is it's way, way out to the side in that direction. Now looking at the betas, so now we've got the PSA betas on the bottom and then the odds ratios for prostate cancer on the y-axis. You can see that there are a number of things that actually are being found to be associated with both. Some that are just associated with PSA and some that are associated with prostate cancer. There is, you look right here at this FGFR, this one's really interesting because they're both associated with increased PSA levels, but one is associated positively with prostate cancer and one is inversely associated with prostate cancer. Those are the kinds of things, like when you're a student, you look for stuff like that because it's like something weird's going on here, or something interesting, I should say. And I don't actually know what, you know, you could talk through different explanations for what might be going on there. I'm not entirely sure. Okay. But basically, what we're seeing is there are indeed variants that you could call PSA variants, not prostate cancer variants. Okay, so the third thing that we did here was to look at the heritability. So what you can do is take your array data from the GWAS array and look at how everyone is related to each other in your genome-wide association study. Because usually you get heritability studies from families, twins, or other closely related individuals. Well, it turns out from the animal literature and the genetics literature of animals, you can actually use REML-based methods to calculate heritability from unrelated individuals. And that's because we actually all are related to some degree. So you can, basically, we could calculate the relationship between everyone in this room, our genetic relationship, and then see whether or not we have similar levels of a particular trait. So you've got your kind of relatedness and your trait variability between individuals, and you can come up with estimates of heritability. So what we found was that the SNPs that were genome-wide significant in our study explained about 7.5% of the heritability in PSA. But you can also look at everything else in the GWAS array. So you don't want to just throw everything else out that's below the dashed line. So you can look across the entire array, and the rest of the SNPs on that array explain about 30% of the variability in PSA. And you add those together, and you're close to what the twin studies, the previous studies were 40 to 45%, and now we're up to 37% just from this GWAS array of the kind of the genetic variability in PSA levels. So there's a debate from these studies, GWAS studies, about missing heritability, and that is that most of these studies have come up, like in this study we found 40 genetic variants that explain 7 to 8% of the variation in PSA levels. Well, everyone threw up their hands and said, well, GWAS are terrible. They're not finding very much of the, there's so much left that's missing, or that's missing heritability. Well, really what this is it's hidden, because it's just the stuff that's not statistically significant. That's explaining all the rest of the variation. And unfortunately, the paradigm in genetics is so testing-based that people just ignore all of that. But when you calculate this chip heritability, you say, no, it's not missing. It's just hidden. You didn't have power to have it cross the threshold, but it's there. And you can, if you increase your sample size, you'll explain more and more. And this is what we've seen in genome-wide association studies. Oh, so the fourth thing that I want to talk about is this idea of, okay, so now we've found a bunch of SNPs that are associated with PSA levels. It looks like many of them are associated specifically with PSA and not prostate cancer, and they explain quite a bit of the variability in PSA levels. Well, okay, now could we actually take a man's PSA and adjust it for his germline genetic variants and come up with a normalized measure that should be used in screening? So what the general idea here is that you're going to calculate for each individual a new PSA measure, so PSA prime, and it's this adjusted. So you're going to come up with an adjustment. You're going to multiply by his actual measured PSA. All right. So let's say my PSA is 3, and then we would look at my germline genetic variants, see whether or not I have more. I'm predicted to have a higher or a lower PSA level than the mean, and adjust me based on that. Okay, so if I have an inherently high PSA level, we're going to actually pull my 3 down to like a 2.5. Because let's say the threshold is 4, we're going to say, you know what, you're not as close to that threshold because you just produce high PSA levels. So the way this formula works is you've got, so A is actually the natural anti-logarithm of the difference between the mean predicted PSA levels in the whole population and an individual's genetically predicted PSA. And so what the genetically predicted PSA is it's just the product of the regression coefficients and an observed genetic marker. Right? So let's say for KLK3, which is the strongest predictor by far here, we would look and see, do I carry those genetic variants? And if I do, carry 1 or 2 copies, then I get this G hat, which is the sum across, the product of the regression coefficient for that KLK3 SNP versus what I carry in that gene. And then the average is just the average of these in your population. So we're going across an individual. So say taking the 40 SNPs that we found and calculating their kind of predicted genetic component of their PSA, and then the average. And I'm just looking at the log PSA. So we actually, I should say all along, so these models were all done on the log. We transformed the PSA to the log PSA. And so what ends up happening is your A is less than 1 if you are a high PSA producer genetically and you have an inherently high PSA. It's greater than 1 when you have a low PSA. And that way your PSA prime either goes up or down based on your genetic variants. If you are a low PSA producer, then we're actually going to bump your PSA measurement up closer to the threshold. You don't have to actually change people's PSAs. You could just move the threshold around as well. Have an individual, this is your threshold. When you cross this threshold, we're going to do a biopsy on you. But John, your threshold is 6, not 4. And it's not until you hit 6 that we're actually even going to worry about your PSA levels. Okay, so how does this work? So we went and looked in the Kaiser data, and the nice thing about getting data from a big place like Kaiser is you can get information about who's had a biopsy and which controls have actually been biopsied for prostate cancer and who don't have any disease because they're kind of have been already checked out as being controls. So this figure is showing here on the x-axis, it's just increasing numbers of SNPs that go into this genetic adjustment. So the very first SNP is just this KLK3 SNP that had that really, really small p-value because that is such a strong predictor of PSA levels. And then KLK plus all of the SNPs that are not associated, the PSA SNPs that aren't associated with prostate cancer, and then all the PSA SNPs, whether or not they're associated with prostate cancer as well. And then we actually went deeper into the Manhattan plot and started taking things that had, say, a p-value less than 10 to the minus 4, 6, and then 5. So you start including more and more variants without worrying about whether or not they're passing this genome-wide threshold for significance. So that's the x-axis. The y-axis here is the percent of individuals who are reclassified. And so we've got 2 different colors, red and blue, and the solid lines indicate whether or not you were classified downward, and the dashed are whether or not you were classified upward. So what you really want is to have more cases get pushed up and more controls get pushed down, because this is the real test of whether or not this reclassification works. So the top line here, these are cases who are being classified downward. So you can see that was a large percent, and these are cases that were being classified upward. Right? Now I have to ask you, does that look good or does that look bad? Right? We've got more cases being classified down. Now maybe I should skip to the controls. We've got more controls being classified down and fewer being classified up. That's good. That's what we want to see. The cases, like, wait a minute. This is not what we want to see. We're like pushing more cases down, their threshold down. And spent quite a while scratching our heads about, like, what is going on here? Why are we, this means that we're going to actually not be screening the cases as often. And then we finally realized what had happened here. When you think of reclassifying people, you have individuals who are, in this case, we did basically an age-adjusted PSA. So it depends on, because this is how Kaiser does it to decide whether or not someone should move on. If you're younger, it's a lower PSA threshold and it increases. The problem is, cases all had high PSA. The cell of having a case who's going to move up was almost empty, because that's how they got diagnosed, was they had high PSAs. So we really need to do this in some European population where they're not screening, because you can't move up when there's nobody in there to move up, right? I mean, the cell count was so low. So we did something a little bit different, and that is we just looked at individuals with Gleason score, and whether or not those with a Gleason score is actually a measure of the tumor cellular differentiation of the tumor in the gland, the prostate gland. So high Gleason score is bad. It means that poor cellular differentiation. And they range between 2 and 10, but mostly are 6, 7, and 8. Right? And so we looked at using a threshold of 7, so whether or not you were above or below 7 Gleason score. And now we have the blue line are people who are below 7, and whether or not they're getting classified up or down. And the red line is people who are above 7, up or down. So these are all cases. And what we're seeing now is that the men with the lower Gleason score are the ones being classified down more. So that's good. That's what we want to see, is we want to say, okay, you have the more latent prostate cancer, so we're going to push you down a little bit to the screen. So for this part of my talk, so basically the idea here is that incorporating genetic information can help us figure out who should be screened and reduce the number of unnecessary biopsies, and among men who are high PSA producers, but increase the number of biopsies for those who are low PSA producers. And the ultimate number of biopsies should be lower, and hopefully the idea is to improve sensitivity or specificity. Now this is actually, for most of my research where we're trying to find risk factors for cancer, this is actually the closest thing I've had to something that could go into the clinic. If this really works, we could start using it immediately to try to improve on this kind of controversial measure of PSA. Okay, so and this is a picture of my lab. I of course want to acknowledge all the funding support for this research, and also Kaiser Permanente, my colleagues there. But I also want to move on to the last teeny bit here and talk a little bit. When I was first invited to give this lecture, I thought it was because I was one of Bob's classmates. And it didn't, so Bob and I actually entered UCLA at the same time. So we were all through our cohort together at UCLA and actually worked on a number of projects together. So I want to tell you a little bit about my experiences with Bob and with that. So he and I worked with Robert Hale on, we were both research assistants in his lab on the premenopausal bilateral breast cancer project. And that was a lot of fun and really interesting. But one day Robert came in and he came to Bob and I specifically, and he said, I've been approached to design a study to look at the health effects of swimming in polluted water. Now this is, I mean, neither of us have done anything in this area since then, but we did this one really big cohort study looking at whether or not swimming in water that's polluted with either fecal matter or it's just close to a storm drain in the Santa Monica Bay increased your risk of eye infection and gastro illnesses. And I still try to wonder, why did Robert have a pretty big lab? And he came to Bob and I because we both like to surf, and we were both really interested in this area of going out in water. So this study ended up, it was a big cohort study. So basically, Bob and I, we were about third year PhD students at the time. And we got to design the study. It was incredible. I mean, we were basically, we brainstormed with Robert, and we came up with this whole idea of how we were going to do this study. And the beaches in LA are crowded in the summer. right? There's a lot of people in beaches. So what we did was we had a team of interviewers. First of all, we put flags up so we could actually mark where people were sitting in the beach, and more importantly, where they were swimming. Because most of the previous studies of the health effects of swimming in water that had storm runoff were comparing swimmers to non-swimmers. And those are different types. So we really wanted people who went in and got their head wet. Everybody had to go in and get their head wet. And what the interviewers did was they went down and basically recruited people on the beach all summer long, and they would note whether they were in front of the storm drain and also where they were relative to these flags that told us how far away from the storm drains they were. And we also went and sampled the water right where they were swimming. And it was actually, so this is the paper we published from it in Epidemiology. It took us quite a while to publish it. We both finished 5 years plus before the paper came out. But it actually, so this paper's been cited 400 times and actually changed the requirements for when water could be called safe to swim in. I mean, like the EPA and the CDC picked up on this. So as far as public health impacts, this was actually a pretty big one for this one project that we did. And I don't know, it was great working with Bob and coming up with the design for this study. But the other thing, Bob was, he was not only a great scientist and a great classmate to have, but he was also a really good friend. And I can tell you the one measure that you can tell whether someone's a really good friend when you're in graduate school is whether they help you move apartments. Okay? And Bob, he had a truck. And he was Bob. He was a big, strong guy. He helped me move twice. Twice. Most people, you ask